Hi everybody, and welcome to the second part of my section of the class. And we're speaking about case studies in science literacy with a focus on genetic engineering, or how we say in a more common parlance, GMOs. And today's lecture will really work around the idea of the currently used suite of transgenic organisms that are commercialized, the ones that are currently available for us to use in different medical as well as food and crop applications. It's important to note that these do span a wide variety of different opportunities across medicine and agriculture. So what we'll talk about today is basically everything that we talked about last week applied. So last week we talked about how you make a transgenic plant, the idea of how do you get a gene in, what happens once that gene is in, and how do you make those selections. Today we'll expand on those concepts. We really have to start out with a little bit of review, because you remember that the basic idea of genetic engineering is to achieve some sort of outcome that you can't do with just standard genetic techniques. So you can't do it with breeding. You couldn't really, well, you could if you had 1,000 years or 10,000 years. You can very quickly make precise changes inside an organism's genome. And the two ways that we do this are we add a gene or add some sort of trait. For our purposes, they're kind of one and the same. You add some sort of gene that confers a specific trait that normally isn't there. The other thing that we'll do is we'll either suppress or even eliminate a gene that we don't want. So between these two mechanisms, adding something there that wasn't or taking something out that is, we can achieve a plant improvement that gives you the trait that you're looking for. So, let's talk about these two things specifically. The first thing we'll talk about is how do you add function? How do you change the expression of a gene that normally wouldn't be there? Or maybe one that is there, but maybe in a related line that isn't in the exact variety where you'd like to see it. The way that we do that, we have to go back to first discussing that central dogma of molecular biology. And we talked about this last week, but here's a quick refresher. In the central dogma of molecular biology, you have this situation that you see on the right, that you have DNA that's tucked away in the nucleus, that a series of factors engage in what's called transcription, copying that DNA into a transient form called RNA that leaves the nucleus, goes out into the cell, at least in a eukaryotic organism, goes out into the nucleus, where it's translated, where the information is turned into a protein. The information is used to make a protein, I should say, that carries the information from the DNA through that RNA intermediate to the final structural and functional element of the cell, the protein. Now on the left, I have written down a little schematic that says promoter, and then a little arrow, and that says your favorite gene. And this is the way in molecular biology how we typically will put out the notation of a gene. What it means is that your favorite gene is all of that information that eventually will end up in the protein. It's the coding sequence. It carries the coded information that represents the series of amino acids that will eventually end up in that protein. But that area before it that says promoter, before that little arrow, that's a regulatory region. It's a region that carries a whole bunch of important DNA sequence that's responsible for turning on your favorite gene, or I should say the gene that's downstream from it, the information that's downstream. In this case, your favorite gene. So if we wanted to add a trait, like let's say a protein that provides some sort of protection from insects, we could take that protein, or the sequence for that protein, your favorite gene, and place it into a background next to a promoter using recombinant DNA technology. We can use a promoter to turn on that gene in a context of a transgenic plant that we talked about last week. But this is the basic schematic here. The other way that we can do this is by suppressing a gene that we would like to turn off. So in that previous example, we could add something to the plant's hard drive to turn it on, to express it when it normally wouldn't be there. What I'll show you next is how we can turn that off. So this is an example of the native gene inside the plant. It has its own native promoter, so its own control region, turning on, say, gene X. But we want to silence gene X. I talked to you last week about a couple ways to do that. One of which ways would be to turn off the RNA or degrade that RNA intermediate. That if we could destabilize it or degrade it, it would stop that information from the plant's hard drive and the DNA and the nucleus from ever being turned into or translated into a protein. So how do we get rid of that RNA intermediate? And there's a couple ways to do that that scientists have discovered. One is to create something called antisense. And it's where in the laboratory you generate a construct, we call this, a scenario where you have a very strong promoter, so the DNA control region turned on at a very high level, with gene X's sequence in the opposite orientation. And essentially what it does is it creates an RNA of gene X that cancels out the native RNA that's produced from the promoter, the native promoter, the piece above. And this may be a little confusing and advanced, but think of it this way, that if a gene is producing some sort of intermediate naturally by the plant or by the animal, that you can add something that would be the exact opposite that would cancel it out. That's the easy way to think of it. We see this even more strongly when we look at the idea of hairpin RNAs, because we can add a strong promoter with something facing forward and backwards that folds back upon itself, as shown in that diagram there and down below. And these kind of hairpin constructs are very potent in suppressing RNA and knocking out that intermediate. And we'll talk more about this later. The whole mechanism seems a little bit curious. Why would an RNA sequence that has the opposite sequence possibly cancel out the original sequence? And what it boils down to is a plant or an animal's surveillance mechanism that it uses to kind of look for unusual situations, usually viruses. They tend to be hallmarks of viruses that would suggest that something's wrong in the cell, and the cell has a mechanism to identify it and degrade it. In this case, we're able to create these kind of hairpin constructs that target the native gene, suppressing it, basically turning it off. In the future, and even we're starting to see now, and we'll show you some good examples next week, of what's called gene editing. And this is the case we briefly discussed last week, where we can make a very specific prescribed deletion or change, a knockout, in our gene that we'd like to suppress. So it's able to make a gene that deletes its function, or maybe just changes it a little bit, but usually knocks out its function. This is what we know as gene editing, and this is another way to stop the function of a gene. So, over the next couple of slides, over the rest of the talk today, I'll show you specific examples that are in place now, where either some sort of function has been added to an organism to turn a gene on that normally isn't there, and then I'll show you some good examples of how we've been able to use antisense and RNAi and RNA interference from the hairpin and gene editing to turn a gene off. Basically, by turning a gene on or turning a gene off in a given genetic background is the basis of being able to do genetic engineering. So what are some of our currently commercialized genetically engineered plants? And we can go back to 1982, 1983, I guess it was, to find the first one. And usually people don't think about this, but this was the first genetically engineered organism and involved a human gene being turned on in a place where it wasn't supposed to be, in bacteria. So scientists were able to obtain the sequence for human insulin. Take that DNA sequence, place it into the background of a bacterium through genetic engineering, recombinant DNA technology, and produce the first product that was being used for fighting a human condition, and also the first genetically engineered product. So the first GMO wasn't a plant, it was a bacterium that made a human protein. This is really important because we have an increasing problem with diabetes. And this was really starting as an uptick in the 1970s. That patients require management of their insulin levels, sometimes by injecting insulin. And there's a couple types of diabetes. Type 1, where you don't make any insulin, so your cells don't produce it in the pancreas. And type 2 is where your body isn't responding properly, but both cases have to be managed by adding exogenous insulin at the proper times. The problem is, where do you get insulin from? Well, starting back in the 1940s and 50s, you would obtain insulin by purifying it from pancreases from slaughterhouses. So you would use pig, cow pancreases to remove the insulin, purify the insulin, and then inject that into people. That's the way that it was first done. It was originally done, it was first identified in 1921 from a dog pancreas, and bovine and porcine, so pig and cow versions, were used to treat the disease or the disorder. The problem is you can see many problems. We'll come up with those in a second. So what's another solution rather than injecting it from pigs and cows? The main problem there is that if you're injecting pig and cow insulin, with the number of diabetics today, is there possibly enough pig and cow pancreases to be able to satisfy that demand and be able to have pure insulin? I mean, what's coming along frequently are not just pig and cow insulin, lots of other proteins that could contribute to allergic responses. So what science did was step up and say, what we'll do is we'll make this thing using recombinant DNA technology. And what they did, and this is an example of the paper from 1979, was to be able to synthesize human insulin in E. coli, in bacteria. So here you could grow giant fermenters of bacteria that would produce 100% pure human insulin. The first real good example of a genetically engineered protein for human therapy. And it's important to remember how this works. That bacteria, so this slide shows a model of a bacterium that reminds us that its genome, all of its genetic material, resides in two places. One, it's inside this circular chromosome, but it's also in these small things called plasmids, which are small circular pieces of DNA. And the small circular pieces of DNA are what we as scientists manipulate in order to add a gene. We can put the information we want onto a plasmid and introduce that to bacteria. And once it goes into bacteria, the bacteria multiply, they multiply the plasmids, and as the bacteria grow and divide, the plasmids grow and divide within them. And every bacterium now has these instructions to make 100% pure human insulin. And I'll show you some other examples with some other proteins in a bit. The next slide shows this as well. This slide shows that you have a human cell where you can obtain the DNA sequence represented by that pink bar. And then we can place that into that blue bar using recombinant DNA technology, basically cutting and pasting that one piece into the bacterial plasmid. Then the bacterial plasmid with the human insert is now placed into bacteria where you can grow vats and vats of bacteria that now express that 100% human genetically engineered insulin. It all starts with being able to obtain the gene out of the human chromosome. And actually it's pretty routine these days. Now that most genes have been sequenced in animals and plants, we understand them very well. It's very easy to, what we say, clone the gene or copy the gene out of the genome and then move that into a bacterial background, into a bacterial vector. In this case, that human insulin gene was moved into the bacterial plasmid and then expressed in giant fermenters. Here it's shown some bacterial medium and then the fermenters below that do nothing but produce massive amounts of human insulin inside bacteria. The nice thing about it, it's 100% human insulin. You can scale up or scale down to meet demands, and you don't have to worry about other cow or pig proteins. There's also some religious oppositions that happen with respect to using animal proteins into the human body, and there were always some questions about could you co-purify some disease. And there's some examples I'll show you from pituitary glands in a moment that suggest that's the case. The problem is we've created a wonderful product that no longer requires animals to be the surrogates, that produce a protein that we can produce in bacteria in amounts much less expensively and with much greater purity. The first genetically engineered organism is one that we see in use all over the world and provides a stable and predictable source of an important life-giving product. But of course, not everybody's happy about that. You know, we have this movement that likes to stand up and say this is dangerous technology. And they've even taken a stand on insulin. So when we start back with and this is from 2014, that they're saying that these are dangerous products, that they're unpredictable, that they're causing problems, based on something that they read in some article that may or may not have had much good information. The problem is here, readers of Green Med Info may feel that this is a legitimate scientific argument, a legitimate medical article, and maybe stay away from genetically engineered insulin. Maybe stop taking their insulin altogether. And this is one of the ways in which kind of our rejection of technology can have some very serious health implications. The same technology was also used to reverse dwarfism. That there are problems with human growth hormone that's produced. In this case, it says somatotropin. That's actually incorrect. I should have fixed that. But human growth hormone is secreted by the pituitary gland and causes elongation growth and longitudinal growth in muscles and bones. So you can see on the bottom, the people there have generally the same proportion of things like head and hands, yet their long bones are not elongating. And the way to fix that is you can use human growth hormone as a therapy to correct it, at least in some degree, at least lead to more muscle growth and bone growth. And the way that you did this was you could purify human growth hormone from cadavers. And you could go into a morgue and with a small bucket collect those pituitary glands, which then you could purify human growth hormone. And 8 to 10 cadavers were needed for a single treatment. So you can see a lot of work, a lot of expense. The other big issue is, again, you're purifying other products, other proteins. And in 1985, there was a paper that studied the incidence of death of people in Great Britain, where 2, probably 4 people died from a rare disease called Creutzfeldt-Jakob disease. It's kind of a human manifestation of something similar to mad cow disease. And it was suggested that this may have come in through growth hormone treatments from the pituitary gland of someone who is diseased. So there are downsides to using, obviously, using human pituitary glands as a source of this material. But once again, genetic engineering to the rescue. That you can take the HGH gene from a human cell, put that into a bacterial plasmid, and now generate bacteria that produce 100% human growth hormone. And it's been shown very, very well that the recombinant form is stable, causes there to be better gains from exercise, better gains from strengthening, and better longitudinal growth. And that, even though the gains aren't huge, they're still pretty good, and suggest that these are important therapies for people suffering from growth or abnormalities. And basically, it's an idea that we can help people who have a genetic affliction that slows the normal growth process grow better with human growth hormone. There's another really interesting one that we don't really talk about, but this is Gaucher disease. And Gaucher disease, or Gaucher disease, is a genetic disorder that eventually causes an accumulation of toxins in the body. It leads to an enlargement of the liver and spleen. It's very common, well, not very common, it's most common amongst Ashkenazi Jews. It's a mutation in a gene called beta glucocerebrosidase. So glucocerebroside is a compound that's toxic to the body and causes these symptoms. And it's detoxified normally by beta glucocerebrosidase. So if you could take the glucocerebrosidase enzyme and place that into, let's say, a plant, and have the plant make glucocerebrosidase enzyme, now you could potentially eat those carrots or eat that plant and then receive the benefit of that enzyme now working inside the body. This was tried, and it was actually done in carrots. And you can actually get carrot cells that are producing glucocerebrosidase, and when you consume them, the structure of the carrot cell, the cell wall, protects the enzyme from being degraded by the gut, by the enzymes in the guts. And a company called Protalix has made this therapeutic that helps people suffering from Gaucher disease. And this is available, it's on the market, it's for a rare genetic disease with a clever genetic solution. So here's an example of recombinant DNA technology, a human enzyme expressed in a plant cell that now allows people with a very rare disease to live normally. Again, a very nice application of genetic engineering technology. Another place that starts to become a little more familiar is in cheese-making enzymes. So in making cheese, and you can see the gentleman on the bottom here is separating the curds from the whey, right? You add milk and you add something called chymosin. And chymosin is an enzyme that causes the milk to curdle, forming the clumps of protein. It used to be that you could you would prepare chymosin, or what they called rennet. You would make a rennet from a mixture of enzymes from the stomach of a calf or from a sheep. and you would add that to milk to create cheese. The problem, again, is you don't have enough stomachs to be able to meet the current demand of cheese, especially because it is best and highest from young cattle. And after people slowed down with eating the veal, there wasn't so much rennet or so much chymosin available to be able to produce cheese enzymes. So the trick to that then became, what if we could clone the gene from chymosin from a calf or from some sort of an animal, place that into a bacterial plasmid or whatever, microbial plasmid, and introduce that back into the organism, and then now have bacteria or maybe fungi do that work for us. Now it turns out that this worked best when genes from a camel, or for the chymosin gene from camel, was introduced into a couple different fungal species, namely Aspergillus niger and this other one, that when those genes from a camel were placed into a fungal background and the fungi were grown in giant fermenters, they would produce much of this enzyme at a very high level. So when you look at different cheeses and you read the labels, you can find examples of cheeses that say it contains no animal rennet, that it's not made from the stomach of a cow. And many people find that important, that they're not contributing to the slaughter of animals to be able to produce cheese. It's a protein source for lacto-vegetarians. And you find even more now, you find now that as people begin to make food choices, they like to maybe stay away from animal products. But here's an animal protein that's made without the animal. So some interesting questions there. But in cheese making, you can buy these gallons of microbial rennets. You can buy this stuff by the gallon. And this is what people use for cheese making these days. And about 90% of consumer cheese is made using a non-animal based rennet, something that comes through genetic engineering. What's particularly interesting is if you think about the recent campaigns we've seen around genetic engineering and aversion to genetic engineering, companies like Chipotle will say, we don't use any GMO material in our food. Well, they do use the cheese, which uses a genetically engineered microbe as an intermediate. It's also important to note that when we talk about the issue of genetic engineering and labeling, which we'll cross in last week of the discussion, Vermont was the first state to pass the GMO labeling law, saying that customers had a right to know what was in their food. However, their biggest product, cheese, was exempt from labeling. Between the Vermont law and what happens at Chipotle, it really makes us scratch our heads about how the intent of this kind of rhetoric. Here's something that's created inside a microbe from 100% recombinant DNA source that's not much different than the plant that makes sugar. We'll talk about that in a bit. But in this case, makes an enzyme that generates, that needed for cheese production. So now we'll shift over to plants. There's really only 8 plants that are happening in production right now: soy, corn, sugar beet, and canola. And those are the only 4 that really end up in your food. That's soy milk, soy solids, corn starch, high fructose corn syrup. From sugar beets, we get sugar, of course. And from canola, usually canola oil. And soy, also soybean oil. Corn, corn oil. So those are the main things that end up mostly in, you can imagine, in processed foods. Papaya from Hawaii and a small acreage of squash are genetically engineered to be virus resistant. We'll talk about that today. And then cotton, which is for fiber, and then some for animal feed. And alfalfa, which is grown for animal feed. Those are the only ones that are genetically engineered, currently commercially available on any major scale. There are a few other exceptions, but this is the major rule. There are really just 3 traits: resistance to insects, resistance to herbicides, and resistance to viruses. So when we think about all of the discussion that's out there on the web about tomatoes with fish genes and strawberries with human genes for reproduction, no, this is it here. This is the entire set of traits and plants that are currently growing them. And next week we'll talk about what are the opportunities that we've lost. This is a very, very small constellation of products, and I think you'd be really surprised to see all the things that we could do if it was only a little bit easier. So we have to do a little bit of review from last week about how we make that transgenic plant. How do you get that gene in there? And we talked last week about this idea of totipotency, about how a plant cell can convert into, start growing into clumps of cells, maybe even turn into a whole leaf that eventually ends up as an entire plant. Just as you can cut the bottom off of a plant at the stem, stick it in water and grow new roots, plant cells can reprogram how they grow. And so this picture here really reminds us about how a single cell from that carrot can be grown into a whole new carrot. And if we could get our gene of interest with the promoter and all that stuff into that first cell at the top of the figure, then we could have a plant that now would contain all the cells containing that new gene that's been introduced. It looks something like this, that there are some new plants coming from a clump of cells that now give us that new plant that contains our gene of interest. And we talked again about the central dogma of molecular biology, about how it really is just permanent information like a hard drive, transient information like a USB drive, and then this printer which can really give us the paper, which would be analogous to the protein. It's this system that allows us different places to turn on or turn off the gene, as I mentioned in the beginning, by adding something new to the hard drive or by getting rid of that USB drive. And it's a good analogy, it works, but this is the basic idea. So let's talk about the places where we have, for commercial purposes, gone ahead and added genes or taken genes out using these mechanisms. Now the first one I'll talk about was the first genetically engineered commercialized plant, even though we currently don't grow it. And this was the Flavor Saver tomato. And this was done in 1994. It was approved in 1997. It was approved in 1994. The problem is that tomatoes, as they gain in flavor, they start to become soft. And you lose the ability to ship them over distance. You lose their post-harvest quality. So as they taste better and better, they become mushier and mushier. So it makes it harder to bring a good tomato to the store. So the trick here that scientists had at Calgene was uncouple the two processes. Make ripening and softening go slower while keeping the biochemistry of flavors and aromas going at the same rate. And the way that you could do that was pretty simple because we understand the genes that are associated with the softening process. And it boils down to an enzyme called polygalactoranase. Polygalactoranase, or PG, is expressed by the plant. You can see here's the schematic of that plant's promoter, so it's regulatory region that turns it on and off, turning on this arrow, which is the gene sequence for PG. And then it makes this stuff, this intermediate called pgRNA. Scientists were able to put in an antisense construct which created this backwards pgRNA. And that pgRNA now creates a situation where the forward and the backwards can combine together to basically cancel each other out. So it looks something like this. The two end up actually physically interacting and igniting a system called RNA interference. And RNA interference, or RNAi, causes the degradation of both of the pieces of RNA into small, tiny little pieces that are then just discarded by the cell. So you've essentially turned off the instructions for the plant's enzyme that causes softening. You took the USB drive that plant was using to communicate information from the nucleus to create new proteins and destroyed it. Hit it with a hammer, run it through the washer. You know, all the things that we do with USB drives. So this is how the Flavor Saver works. By using RNAi, or this RNA interference intermediate process to degrade the information that was required for the production of the enzyme. So it worked pretty well. The big problem is that the tomato that it was in didn't taste very good in the first place, so there really wasn't much of an improvement. The lines that were transformed with this construct didn't taste great, and there was no value for the consumer. Therefore, the company wasn't making much of a profit. So a good idea on paper for people who really like tomatoes, but poor execution. And it ended up going away. That went off the market. But let's talk about some things that stuck around. Some other examples were very good. And one thing that you frequently see online is that genetically engineered crops have been engineered to create pesticides. And you see a lot of imagery like this, which is meant to scare. Of course, it's important to keep in mind that plants produce lots of compounds to defend themselves against their predators, against bacteria, against fungus, against insects. Plants produce lots of compounds to take care of these things. It's important to remember because 99.9% of the anti-pest compounds you consume occur naturally in plants. So nicotine, caffeine, pyrethrum, many other volatile compounds like linalool, methylanthranilate, all these other compounds are made naturally by the plant and have roles in deterring insects or deterring fungus or bacteria. Plants are pretty vulnerable. They're stuck in one place. They need to make chemical defenses. So there's a lot of good examples in throughout biology where organisms make a product that affects some other kind of pest or some other kind of pathogen. We'll focus on one of those today, one called Bt, named after Bacillus thuringiensis, the bacterium it comes from. And this busy slide shows a whole number of different antimicrobial, anti-nematode, antibacterial, antifungal compounds that plants make. One of these is shown here in the circle. It's called a cry-toxin, which is nothing more scarier than the cry-toxin. Really what it stands for is it's a crystallized protein, cry, crystal, and it is a toxin called delta endotoxin. It's a very toxic compound if you're a moth larvae. And if you choose to chew on corn or other types of crops, one common one is cabbage loopers, so they live on brassicas. Their larvae, so the caterpillar stage, when it consumes Bt, this crystalline protein, it gets sick and it dies. This has been known for a long time and has been a huge success in organic cultivation. That you can buy this stuff called dipel, and if you read the label with dipel really carefully, it says made with crystals or spores from Bacillus thuringiensis, containing the Bacillus thuringiensis toxin called Bt. So Bt has been very, very useful in organic cultivation, allowing people to farm without having to spray organophosphates or carbamates or other types of pesticides that work neurotoxically on the insect and are non-selective. They kill all insects or a large majority of insects. Bt just targets the one species or the one very closely related species of target. How it works is pretty remarkable. You can sprinkle Bt spores or Bt spores on the plant. So it's shown on the top here where that caterpillar is eating them. And inside the gut of the caterpillar, it binds very specifically to a receptor that's only found in the gut of that caterpillar. So the receptors are like a lock and key mechanism, that they bind the protein and then they cluster together to form a pore, which then allows the inside of the body and the rest of the body to intermingle, that you have a mixing of the stomach contacts with the rest of the body, and the animal gets sick and dies. So just a few little nibbles of that leaf with some Bt causes this organism to become permanently turned off to it. Kills the insect too, so it doesn't reproduce. So scientists thought, well, what if we were to take the same information to make that Bt protein, that delta endotoxin, and place it into the corn, place it into cotton. Now if you place it into the plant can make the protein and protect itself. And this is exactly what happened. You're able to make plants that can protect themselves from predators. And this was really helpful because the mechanism was so specific that it worked only on the ones it was targeted for, that you didn't have to spray an entire field and kill all the insects, including the beneficials. That now you're able to target only the organisms that were chewing on the plant. Back in the 1980s, I thought this was brilliant. And I thought this was going to be a huge success for farmers because everyone will love this. We're cutting insect use. We're allowing farmers to grow plants that protect themselves. And this seemed to me to be the way that everything would be done as we went forward. You see some of the major ways in which this works. That here's an example of corn that's protected and corn that's not. Cotton that's protected and cotton that's not. So you can see in the center there the non-GM cotton. And on the bottom, the eggplant. We'll talk more about that later. The advantage to this is that you've seen a huge decrease in broad-spectrum insecticide use. It's something like 300,000 tons of broad-spectrum insecticide use on cotton in China in 2003, not used because of the Bt. It lowers fuel and farm costs for farmers and really had good dividends in the developing world, and again, no effect on beneficial organisms. The limitation is that you do see pockets of resistance that are occurring, that you find places where the insects are developing and evolving around the Bt toxin. It means that farmers do have to go back to more intensive scouting and use of old school chemistry to be able to control them. The next major example of genetically engineered crop is the glyphosate-resistant or Roundup Ready products. And what this does is it allows the plant to be sprayed with a mild herbicide that kills the plant, but is relatively harmless to animals. And if you look at this picture, it shows some corn that's growing through some soybeans, where the corn was not Roundup Ready, but the soybeans are. So what does Roundup Ready mean? What is glyphosate? Well, glyphosate is a rather interesting molecule. Glyphosate is the basis of the world's most widely used herbicides. They become very popular because they're inexpensive, they're very potent, and they're relatively harmless to humans. So what you're looking at here in this top panel is the chemistry that it catalyzes. It works in amino acid synthesis. So the things that are the basis for protein synthesis, the basis for proteins. Proteins are made of amino acids. There are specific amino acids that require this pathway in plants. That thing on the one side, that shikimate 3-phosphate, is combined with elements from this phosphoenol pyruvate to be able to create this other thing, 5-enol, vinyl, whatever that says. So you create this product that then is converted into aromatic amino acids. On the bottom, what you're looking at is where glyphosate binds into this enzyme. So A is this enzyme that catalyzes the reaction. B is that complex bound with glyphosate and shikimate 3-phosphate. Shikimate 3-phosphate, glyphosate is basically competing for that space that phosphoenol pyruvate would take. And when glyphosate's in that space, the enzyme doesn't work. So it's a way of stopping the amino acid production because you're binding this herbicide into this critical enzyme. It works on all plants, except for the plants that become resistant. We'll talk about that in a moment. But the pathway isn't present in animals, which is why it has such low acute and chronic toxicity. In any estimation you see online, if you look at what the relative toxicity is, it's reasonably low. And there's plenty of things that show that relative to other compounds. You see Roundup there on the left as the third or fourth thing in. It's about as toxic as table salt acutely, and much less than something like, say, Advil. Last year, the IARC classified it as a probable carcinogen, a very, very controversial decision based upon a couple of data points that maybe weren't the best data points. And I think when you look at all of the other world's organizations that classify carcinogens, they did not see this as a carcinogenic problem. But how does it work? Well, one of the other aspects of this, I told you how it worked. Let's talk about how it's used. Farmers spray this, so you plant your seeds, you let the crop grow, let the weeds grow at the same time, and then spray over the top with a single dose of Roundup, or maybe other ones later, but one dose of Roundup that then kills all of the non-transgenic plants, leaves your crop standing, kills all the weeds. And you do this at the first trifoliate leaf stage. So you're not spraying it on food, you're not spraying it on the items that will be go into processing. You spray it on the plant at a young age, and you use about 750 milliliters per acre, or about 25 fluid ounces. So you're using about 2 soda cans of the active ingredient over an acre. And what you're seeing there on the right is mostly water. This is a sprayer that's loaded up with water that's distributing glyphosate on some soybeans. So briefly how it works again, and this is important. It's really important. I neglected to mention earlier that there is a bacterial version of the same pathway, but the bacterial form does not bind the glyphosate molecule. That's really important, and I'll show you why here. So this is that pathway, that A to B to C makes amino acids which go into proteins, and that one step that's catalyzed by EPSPS, by that enzyme I spoke of earlier. It turns B in the sea. Glyphosate binds into the EPSPS protein. When glyphosate binds into EPSPS protein, it no longer can function, and that plant dies as the biochemistry has been disrupted. We know that bacteria have a comparable parallel system, only that enzyme is a little bit different and doesn't bind glyphosate. So now you can see the obvious solution. What if we were to take the bacterial form, put it downstream of a good promoter, right, downstream of a good control region, and place that bacterial version into the plant? Now the plant has this bacterial version that can't bind glyphosate, and so no matter how much you spray, that plant will survive. This is the way in which glyphosate resistant works. It's basically switching out the one form of the enzyme that does not bind the herbicide for the one that does, and allows the plant to be resistant. The problem is that when you have a technology that works well, such as glyphosate resistance, and it's the only technology that's been approved from genetic engineering and herbicides, lots of farmers use it. When you overuse anything, you start to see the downside. And we'll start with the limitations. That you see weeds evolve rapid resistance, and it means that you need increased labor, you need more hand weeding, yields go down, you need newer chemistries, you need new ways to be able to work your way around it, sometimes going back to old school chemistries. That heat map on the bottom shows the increasing number of resistant weed species as you go from cool colors to hot colors. That glyphosate resistance is a major problem in some areas, and the weeds are taking over and now require new ways to solve that problem. The advantages are still there, and farmers still do use the technologies in many ways. Again, low toxicity, cheap, that it really saves farm and labor costs for the farmers and fuel costs. The big advantage is decreased tilling. Farmers don't have to turn the soil to get rid of weeds because you can spray a chemical on the top. It allows farmers to keep their topsoil. You have less erosion. You have better establishment of organic material in that topsoil layer. So a lot of advantages, some definite limitations, and it's something for us to keep in mind whenever we talk about these technologies. The last one I'll wrap up with today is a really important one that again talks about disrupting the RNA. RNA interference that breaks down this transient information in the middle. Only the hard drive was not the nucleus of the cell. The hard drive was a virus. In Hawaii, the papaya ring spot virus was causing massive problems in the 1970s and 80s. And it was causing tremendous tree decline as well as unsaleable fruit that were a traditional and important component of the diet in Hawaii. The way to beat the virus? Using RNA interference. And this slide shows the same process again, that you had the viral promoter turning on, oh, I didn't change the name of the gene. Let me see if the next slide does it. There it is. I left the wrong slide in. You have the viral promoter producing something called viral coat protein. And so if you were to add a gene that had the viral coat protein sequence in backwards, the two would cancel each other out and be degraded and provide some level of protection because a virus can't live without its coat protein. It worked like a charm. And when you looked at trees in Hawaii, you would see the difference. So the ones on the left on the top panel are not protected, whereas the ones on the right are genetically engineered to resist the virus. On the bottom, you see the opposite, that you can tell they're resistant from the non-resistant trees. And this is the first time I've really discussed the podcast called Talking Biotech, where in episode, I think 26, the scientist that created the transgenic papaya was interviewed, and it was a beautiful discussion. Here are two infected trees, one that's protected, one that's not, in a field of protected trees versus those that aren't. This was a great example of how you could use RNA interference, basically breaking the thumb drive of that process to be able to create trees that would protect themselves from disease. So to conclude, we know that the products from genetic engineering have been used by humans in health and food since 1983, starting with insulin and human growth hormone, and finishing with more modern examples in food where we can see advantages of insect, herbicide, and virus resistance that we're seeing in a variety of different crops. Right now, the current number of crops is reasonably confined just partially because of a regulatory system that is slow. And we'll talk about that more in week 4. Most of all, it's important for us to note that there have been 0 instances of health problems that have come from the technology itself, that there have been 0 instances of food or of medicine that have caused problems because of a genetic engineering. We also are cognizant of those environmental problems, such as the resistance to disease, resistance to insects and resistance to weeds, that do threaten how well these technologies will work going forward. They require new solutions and more clever ways to solve the problems. Again, my information is here on the slide. Please reach out if there's any questions. I'll be happy to answer those. And most of all, it's important to maybe listen to the podcast if you're interested in these topics. We talk about the advantages in food and medicine and the ways in which they're used to solve problems for people and the planet. Next week, we'll talk about the examples of genetic engineering that we don't use. This week, the things that have been implemented. But next week, we'll talk about a huge number of solutions that are on the shelf and sadly can't be used. In week 4, we'll talk about why.